Well, good afternoon, everyone. I'd like to welcome you to today's edition of the HNRCA Monday Seminar Series. I'm Dr. Roger Fielding. I'm the Associate Center Director here at the HNRCA. It's my pleasure to introduce Dr. Ben Miller from the Aging and Metabolism Research Program at the Oklahoma Medical Research Foundation and the Oklahoma Nathan Schock Center. Dr. Miller received his Ph.D. from the University of California at Berkeley and did postdoctoral work at the Institute of Sports Medicine in Copenhagen, Denmark. For many years now, Dr. Miller has been a leader in the field of using novel stable isotope approaches to study proteostasis and the implications of that system with aging. So Dr. Miller's talk is entitled Using Stable Isotopes to Understand Proteostatic Maintenance in Health and Disease. And I just want to welcome Dr. Miller to our seminar today. So take it away, Dr. Miller.
All right. Thank you very much. And let me see if I can share my screen here. Oops, I think that might be the, do you have my presentation there?
Just put it in the presentation format.
Okay, and making sure you don't have my notes page.
All good.
Okay. All right, thank you very much, Roger. I really appreciate having this opportunity. As Roger mentioned, I'm going to talk to you a bit about stable isotopes today to understand proteostatic maintenance. In my lab, we are called the Translational Biogerontology Lab. So I really study the interaction of stress resistance, protein turnover, and mitochondrial energetics. I think these things are related upon each other, so they're really dependent on each other. You can't really have stress resistance without turning over the proteins to adapt to the stress. You need energy to make those proteins, and also that mitochondrial energetics relies on having functional proteins for those processes. So I see these as all interdependent on each other. And we do this in the context of studying the aging process and how to slow that aging process. So my focus over the years has been primarily on skeletal muscle. We're doing a little bit more in the brain now, like a lot of people, but and we look at a number of other tissues as well, but mostly focused on skeletal muscle. As Roger mentioned, we use stable isotopes, and it's really become a specialty of the lab. I take a pride in the fact that we are very translational, that we take things from the dish through animals and into clinical trials in humans. We're really focused on exercise and pharmaceuticals. And again, this integrative approach in our science. So many of you will be familiar with this if you study the aging process. Let me just get a pointer here. So we are really interested in the health span, this period spent free of chronic disease. And I think this definition of health span is one that's continuing to evolve, but it's one that we use. The version of it that we use is that we are free of chronic disease. So once you start to accumulate chronic diseases, you're no longer in the health span. So with treatments and with healthcare, we've become very good at prolonging lifespan, but we've also become very good at prolonging this period spent treating chronic diseases. And in our lab, we're really interested in how to prolong this period of health span in order to compress the period spent with disease. So we're really interested in adding quality to the years rather than quantity to the years. So I'm going to launch right into a little bit of a story here. And this story stems from the fact that there's a lot of very good reasons to use transcript as an outcome in studies. What I've noticed in the data quite a bit is when people use transcript to infer protein from those measures. But we know very well that there's often quite a disagreement between those two. So some recent studies have shown quite well when now that we've been able to add large scale sort of proteomic techniques to catch up with some of the RNA techniques. When looking at heart and say skeletal muscle within a same sample, so this is the same sample in the heart being analyzed with RNA-seq and with proteomics. And in heart and skeletal muscle, you see very poor, about 0.5 to 0.46 agreement between those values. And if you look at the change in RNA versus the change in protein, say with age, you see that this agreement falls apart even more, so that these relationships are quite dismal. So we cannot predict, what this is saying is we cannot predict the change in protein from a change in RNA. And if we look at other more recent data, looking at the difference between RNA and protein for young and older mice, you can see again, confirming that with aging, what we're interested in my lab, this agreement between RNA and protein falls apart quite a bit. So what are the reasons why those don't agree quite often? And the most obvious one is that there's significant post-transcriptional regulation. Now, quite a while ago, we published a paper because we were interested in the fact that many papers were using PGC1 alpha as an endpoint, basically, to say that there is mitochondrial biogenesis. So we know that this co-transcription factor goes to the nucleus and turns on, through other proteins, the transcription of mRNA that are related to mitochondrial biogenesis. So this is called the master regulator of mitochondrial biogenesis. But we also know that things that activate PGC1 alpha, such as things like caloric restriction or even exercise, they also inhibit mTOR. And mTOR is, your primary protein involved in the translation of proteins. So measuring the activation of this, or even the mRNA that result, ignores the fact that many of these treatments are simultaneously inhibiting translation. So we don't actually know if those proteins are ever made. So and besides, translation, there's significant number of post-transcriptional regulation that might impact whether mRNA is actually being made into protein. The second reason is that I'd like to highlight is what I refer to as snapshots. So if I go downtown New York City and take a picture within this very beautiful building, and I come back 6 months later and I take another picture, it appears that not much has changed over this period of time. And in physiology, we're often limited to snapshots, whether that is taking a sample from a petri dish, sacking an animal at a particular point in time, taking a biopsy. So we get these snapshots or windows into what is happening at that very particular point in time. In reality, what we know is that there is a lot happening over those periods, in between those two snapshots. And what we're interested in my lab is how to capture some of this movement, this dynamics that happens between those two snapshots. We know when we're talking about aging and in this progression of young to old, it is not unusual to see, a study where they'll come in and take a snapshot of the young and the old, say in transcript, and call that an aging related effect. But we know that these genes take different trajectories over that period of time. So they can go up and down. They're not all just linearly increased or decreased between young and old. And we also know that within a day or within a week, there's these cycles of gene transcription. So if you consider something like the circadian rhythm, if I came in and took a snapshot here during the day, it would be very different than the snapshot I took at this point during the day. So when we sort of sum all of those things and we look at, say, transcription of gene X and Y, and we take our sample here versus a snapshot here, we get very different responses. And then if you try to match that up to some sort of protein response over that same 24 hours, those two things don't often match up. And this is a quote from a paper quite some time ago, probably 25, 30 years ago. that, recognizes the fact that physiology are dynamic, integrated, and periodic, the processes are, and therefore it is difficult to define it by looking at a single time point or a single process. So the third point I want to illustrate for this disagreement we often see, if we look at something, through the aging process and through classic physiology, we've learned that, you have a stimulus, you have a receptor that senses it, a control center that compares it, and then it makes an adjustment. So this is a classic negative feedback loop. But often if we have a snapshot and say we have a young active individual or an old individual, and we're studying muscle and we see a muscle loss with age, and we then look at the expression of gene X and say it increases with age, we don't actually know if this increase is the cause, which is often the conclusion, it's driving that change, or we don't know if it's a compensatory change to adapt to other things that have happened. So this paper that came out about 10 years ago now modeled all these contributions of transcription, or how well transcription and protein content and some of these other factors, how good they are predicting protein content. And it concluded that mRNA explains about 40% of the variability in protein levels. And this one identified translation efficiency as a single best predictor of protein. And this is why we've why one of the reasons that we decided to measure protein translation, or the actual synthesis of proteins, as a primary outcome in our laboratory. So to summarize that, mRNA is not indicative of protein. It's very good perhaps for many other things. And then stable isotopes can help determine mechanisms of proteostatic control. And so now I'm introducing this word of proteostasis, which is really just protein homeostasis. And the important thing here is that it's a network. It's the synthesis, degradation, folding, all of these things that regulate protein quality over time. And these three things are really dependent on each other. If you change one of them, it's likely to change the other two. So when we think about turnover versus concentration, I think concentration by itself isn't a great indicator of proteostasis. So if we have 100 of protein X and we come back, we measure it again, and we still have 100 proteins of that protein, it could be that during that period of time between our measurements, we made 10 and we broke down 10, or we made 100 and broke down 100. To me, those are very different proteostatic processes, but our protein concentration wouldn't have told us any of that. And, if we go further with this and say the amount of protein goes up by 10, we can't really tell just based on concentration whether that was because the making of proteins went up by 10 or the breaking down went down by 10. And all of those are really important to understand as a proteostatic mechanisms. So that's why we introduce tracers to our work. Now, the idea with aging is fairly simplistic. With age, we accumulate damage within proteins. And this is because our proteins are exposed to things such as reactive oxygen species, AGEs, cross-bridging, which expose them to damage. The simple idea is, since proteins cannot enzymatically repair, or they have a very small capacity to enzymatically repair, the only way you can so-called repair proteins is by breaking them down and replacing them with new ones. So in a case where we have fast protein turnover, we can probably minimize the amount of protein damage. In the cases of slow protein turnover, these damaged proteins accumulate with time. Now, what I'll build in the rest of the talk is it might not be as simple as this schematic leads us to believe. So if we look at the field about what is the current consensus about protein turnover and aging, it's really quite a mess. So the consensus is that aging slows protein turnover, and this results in the accumulation of damaged proteins. That's what I just showed on the previous slide. But we also have a lot of data that supports that treatments that slow the aging process, like rapamycin or caloric restriction, slow protein synthesis. So both aging and both treatments that slow aging decrease protein synthesis, which to me is difficult to wrap my head around. We also know that, or the consensus is that treatments that slow aging also increase protein breakdown. So things like autophagy, we think treatments like rapamycin and CR increase autophagy. So, but if we go to the literature and look at some of the data that are available out there, this is a very nice study looking at mitochondrial proteins in young control, young caloric restricted, old control, old caloric restriction. And on the x-axis here, we have protein half-lives. So with caloric restriction, it shows that the half-life of proteins get longer. So what they did actually in this study is measure the synthesis of rate of proteins using tracer approaches and assume that synthesis and breakdown were equal, that they're in a steady state. So that's how they calculate half-lives using synthesis data. So if we think about what we said over here, this is showing that protein synthesis is slower with aging, but it also is saying that protein breakdown is slower with this treatment that slows aging, sorry. So this kind of goes against this idea that treatments that slow the aging process increase protein breakdown because the half-lives are actually getting longer. So there are lots of data in the literature that sort of, when you think about them, contradict what is taken as the status quo in the literature right now regarding protein turnover. So obviously there's more nuance than these consensus statements get across. And these nuances are important to understand the aging process and to appropriately target them. So what we use in my lab is deuterium oxide quite a bit. So deuterium oxide is this heavy isotope of water. We use it because it's very easy to use and it's relatively cheap as well. We can use it in media if we're doing in vitro studies. We can give it to humans as glasses of water. We can put it in the drinking water of our rodents. So what happens with the deuterium, through metabolic exchange, it gets incorporated into what become our precursor species. So for bulk protein synthesis, it's alanine that we use, although for some of the other measurements I'll show you, it's any amino acid. We use ribose as the precursor for RNA to look at RNA biogenesis. And we use deoxyribose as the precursor for DNA synthesis. And this becomes our measurement of cell proliferation. So the consensus, or what I see in the literature quite a bit, is that aging decreases muscle protein synthesis. And this statement is so prevalent that it actually is in the literature without a citation. It's just a given. And so when I started to try to track back where this idea came from, it took me quite a while to find what were the data that supported that aging decreases muscle protein synthesis. And most of this is because, like I said, it's just not even cited. So if I trace it back, I think it's because of these two studies, which were very nice studies done quite a while ago now, where they showed that by middle age, the synthesis rates of the myofibers in skeletal muscle or the mitochondria in skeletal muscle were decreased, and this carried out through old age as well. When you look closely at these studies, they're really based on a low N, okay? But this is what we've hung our hats on, basically, is that to tell us that aging decreases muscle protein synthesis. Now, if you look the way these studies were done, which is the same way I used to do a lot of my studies, is they were done with a 6 to 8 hour or 4 to 8 hour infusion of a stably isotope amino acid. And you have these infusions, sampling, and then about 4 to 6 hours later, you take a muscle biopsy and you look at the rate of incorporation of tracer into that biopsy. So the product enrichment divided by the precursor enrichment over time. And I just want you to remember that for a second. When we did studies using rodent models looking at aging related effect, in 3 muscles that we looked at, in older muscle, and I'll tell you that there's about a switch from 24 months that act like adult and 28 act like old, there's actually an increase in protein synthesis. And when we ask ourselves, why are we getting completely the different result as these previous studies did, we have to think about the approaches that were used. Now, when we have since repeated this a number of times, this wasn't just a one-off, but we consistently see that aging increases protein synthesis in the muscle. So the reason this is I think methodological. So if we break down, when we take a biopsy from muscle or we take a muscle sample from a rodent, muscle is not just a muscle protein. There's hundreds and thousands of proteins that make up that synthesis rate that we measure in a muscle homogenate. Now, if we model that and simply think about two proteins contributing to a total synthesis, this is fraction nu over time. And if we consider a fast protein, there's fast proteins and slow proteins, and that the sum of these equal a synthesis rate that we observe in our tissue. Now, if we model this and say do something like a 4 to 6 hour infusion, and we take our sample here, you'd notice that the sum of the proteins is almost completely dominated by the fast proteins. And if we go out further, like we do with our long-term labeling, you see that as we go out further, the contribution of the slower proteins increase. And this makes intuitive sense as well, that early in the labeling period, the fast proteins are the ones actually incorporating the label, and it takes a longer period of time to incorporate some of the slower proteins. So I think this actually contributes to the discrepancy that we see between our measurements and some of the earlier measurements, is because we're capturing a lot more of the slower proteins, which is where you'll find a lot of your myofibrillar proteins, or the ones that give it protein mass. Now, there's in the literature, a lot of people like to rely on this puromycin or sunset method to measure protein synthesis in the muscle. This is done because it's quite easy. It's like a Western blot. But I'll issue some caution here that this measurement is usually made over 20 minutes. And if we look at the gel of where we expect to see myofibers, you can see almost a complete absence of myofibers incorporating the label. So even though this particular study was interested in protein mass and an intervention that regulates protein mass, they actually completely missed the proteins they were interested in due to this short labeling period. So I do issue some caution when using these short-term labeling methods in a tissue like muscle. So again, tissues are complex. And if we think about muscle, muscle isn't just a muscle. Even though it's dominated by the myofiber, which is a multinucleated cell, there's other multiple cell types, mononuclear cell types in muscle as well. So when we take a tissue sample, we're getting multiple cell types in that tissue. And like I said, within a sample, there are abundant proteins, fast proteins, and slower proteins, and less abundant proteins. And what we're getting is the sum average of all of those proteins in all of the different cell types. So what we have started to do is think more about this heterogeneity and whether this can explain some of the discrepancies that we see. So in a previous study, when we looked at skeletal muscle in a control condition and a rapamycin-treated condition, we see what others have reported in that rapamycin decreases protein synthesis. In the mitochondrial fraction, though, we saw that there was not a significant decrease, and it was somewhat preserved. However, now we've developed methods that we're able to look at the tracer incorporation into individual proteins. And we do this using a combined tracer and proteomic approach, which is quite complicated. But if we do that, and I draw a line of unity, and each of these dots are an individual protein, if we look within this mitochondrial fraction that indicated no difference, we see that roughly half the proteins are increased with rapamycin and half are decreased. And if we look at this other fraction, even though overall the rates were down with rapamycin treatment, we still see a significant number of proteins that are increased with rapamycin treatment. So the conclusion that, say, rapamycin or caloric restriction or whatever decreases muscle protein synthesis it's really only true for about half the proteins in this case. And with our proteomics technique, we can look further into other fractions, which I will not get into these particular ones right now, but I do want to highlight some other ways that it can be helpful. So if we look at ribosomal proteins and we look at proteasomal proteins, these are what I like to refer to as the proteins that regulate protein turnover. So this is the turnover of proteins that regulate protein turnover. I added another group here. The groups are not super important for you to understand: the control, rapamycin, and rapamycin plus metformin. But if you look at these average differences, you see in both cases in the ribosomal and proteasomal proteins, we see sort of a similar trend here. But what I want to point out is the regulation of these are much more complex than we can gather from those data. So if we, this is the same y-axis on both of these, and each dot represents an individual protein. What we don't see from looking at data like these is that it appears that the ribosomal proteins are regulated together because of this really tight clustering of the synthesis rates of all the individual proteins, whereas the proteasomal proteins are more spread out. So these sort of proteomic type data are giving us a lot more insight than just looking at muscle as muscle and breaking it down into individual proteins. Now, I showed you before the fact that there's mononuclear cells in skeletal muscle as well. And this is a point we've made quite a few times in the literature, but as of right now, we're still the only ones considering this. So within a tissue, you have these cells that replicate and cells like myofibers that are post-mitotic and thought not to replicate. So every time a cell replicates or doubles, it has to double its protein mass. So the act of cell proliferation by itself requires an increase in protein synthesis to double that protein mass. So when we do measurements, say, of control and rapamycin, again, looking at that mitochondrial protein fraction, if we just look at the mitochondrial protein synthesis, we would conclude that rapamycin does not change mitochondrial protein synthesis. However, what we see in this situation is that rapamycin also decreases the replication, the DNA synthesis, so the number of cells that are replicating. And this makes sense because rapamycin is a well-known cell cycle regulator. And if you inhibit mTOR, you are decreasing the rate, you're decreasing that proliferation rate. And it's no mystery that mTOR regulates both protein synthesis and cell cycle because those two things go hand in hand often. So we created this ratio of mitochondrial protein synthesis to DNA synthesis to sort of account for the fact that a lot of this protein synthesis is just going to cell replication. And if you sort of take cell replication out of the equation, we would see that how much of the protein synthesis is going towards maintaining the existing proteome, which we call somatic maintenance, actually increases with rapamycin treatment. So if our ratio was correct in what it's showing us, we would predict that in a cell type that has rapid proliferation, that almost all of that protein synthesis is simply going for cell proliferation. So recently we tested this and we looked at a tumor, the protein synthesis and DNA synthesis rates within a tumor. And you see that these DNA synthesis rates are really high in a tumor, order, quite high compared to all these other tissues that we looked at. And again, and as we predicted, the ratio of protein synthesis to DNA synthesis within the tumor equated 1. So as we would predict, all of the protein synthesis is simply going for cell replication in this case. So there's sort of a validation of that this ratio that we're using is doing what we think it's doing. So how do we address this complexity then? If we know that different cell types are replicating and having synthesis just for replication, whereas other cell types are not replicating, and we have protein synthesis and DNA synthesis, so how can we capture all that within a tissue? And to do that, we've developed a model recently where we use this mouse called the NuTrap mouse. And what the NuTrap mouse allows for is when you cross it with a tissue-specific Cree, in this case, I'm indicating PAX7, which is satellite cells, is it allows us to label the nuclei with M-cherry or biotin, and the ribosoma proteins are lit up as GFP. So what this now allows us to do is by using isolation techniques for the GFP or the M-cherry, we can isolate the cell. This GFP signal is bright enough that we can isolate the whole cell. And then we take those cells and we analyze them for protein turnover. And in this case, we're doing individual protein turnover. And then we can use this signal to isolate the nuclei to look at cell proliferation by GCMS. So what we're getting then is cell type specific individual protein synthesis and replication, so we can account for all the factors that go into maintaining the different cell types within a tissue. So right now we're doing an exercise intervention using this. Like I said, we have PAX7. We also have some other cell types in the muscle, and we're actually doing this in the brain as well. Different cell types like nuclei and astrocytes and glial cells. So another idea I'd like to introduce to you that might help explain some of these discrepancies we see with aging is that with age, proteins become resistant to turnover. And what I mean by that is that proteins can aggregate, become cross-linked, or do other things that make it exit the pool of proteins turning over. And this is rarely, if ever, accounted for in studies using tracers. So the way we've developed to account for this is using a time course study. So if we do a time course and we look at the fraction nu of proteins over time, the assumption in tracer research is that all proteins eventually become 100% nu. So we have this rise in fraction nu over time, and then it'll reach 100% nu and plateau. What we started to see in that some of our tissue types is that we may not ever get to 100%. We may say plateau at 60%. And when we plateau at 60%, what this tells us is that 40% of the proteins are not turning over. So we thought this might be a good way to address some questions related to proteostasis. So we recently designed a study where we did a time course in young or adult and old animals and looked at skeletal muscle. And when we did this and we looked at collagen within muscle in two different muscles, we see these plateaus occurring at far less than 100%. So in adult muscle, shockingly, only about 28% of the collagen protein is actually turning over, and this gets worse with age. So we termed the pool of proteins that are turning over the dynamic protein pool, whereas those that are not are simply inactive. And you can see how small of a percentage of the proteins in skeletal muscle collagen are actually turning over, and it gets worse with age. And with TA, you can see these values are higher, but still far less than 100%. So we became really interested in this pool of proteins that are not turning over and whether this is indicative of decreased proteostatic mechanisms. So when we looked at, say, lysyl oxidase, which is the enzyme of cross-linking, as predicted, it went up with old. The AGE's accumulation is increased in the old. Protein carbonyls are increased in the old. And then when we did a solubility assay, pepsin soluble and pepsin insoluble, we see what we thought was going to happen is that the proteins that are soluble with age decrease and the ones that are insoluble increase. And we are able to measure synthesis rates in these different pools as well. But all of these factors tell us that these proteins that are not turning over are accumulating damage or becoming insoluble and exiting the pool of proteins that turn over and are potentially indicative of a decrease in proteostasis. So the fact that the assumption is made that protein synthesis, eventually 100% of the proteins turn over, actually impacts the way we need to design experiments and when looking at a protein that perhaps does not go to 100%. So if we look at collagen and I fit a line to this curve so that the line goes to 100%, we make the assumption that it does go to 100%, which I show here. We see that this line does not fit these data very well at all. It's a very poor fit. And we see that the conclusion based from this line was that adult has higher collagen synthesis rates than old if we calculate the slope of that line. But if we instead allow the plateau to go where it's supposed to go by fitting it with this curvy linear line, we see that this, as the data I showed you previously, this plateau decreases quite a bit, but the rate of synthesis to reach that plateau is exactly the opposite of what we saw if we predicted that it went to 100%. So whether we consider this plateau or not makes a really big difference in our calculated synthesis rates. And if we do a traditional experiment where we take one snapshot or one data point to indicate what our synthesis rate of these collagens would be, you can see that if we took it early at day 15, we would have made the conclusion that collagen synthesis rates were not different between young and old. But if we did it out at 60 days, we would see that the collagen synthesis rates are quite different. And different than when we measured at 15 days. And it's all the result of this plateau value that's far less than 100%. So it's really important in tissues or tissue types that may not go to 100% that we measure these things appropriately. And quite honestly, with collagen, I won't do it any other approach besides this time course approach now, knowing that what I've seen from the data we've collected so far. The other assumption made in protein turnover studies is often that you're in a steady state. Now, what a steady state means is that the measured process stays constant over the period of measurement. And this is a relatively safe assumption when we are using 4-hour amino acid infusions. We don't expect a large change in protein mass over 4 hours. But now that we've used deuterium oxide and we're going out days to weeks, this is not as safe of an assumption anymore. We often are interested in measuring, say, a period of muscle loss or muscle gain. And therefore, we are purposely not in a steady state because we're gaining or losing muscle mass. So I pulled a study from the literature that was looking at a denervation study, a period of muscle loss. This is to examine some issues related with myofibrillar protein loss. And you can see when they looked at the TA muscle, they definitely lose muscle mass as the denervation model would, for exactly the reason you use the denervation model. In this study, they just measured FSR as we traditionally would and saw that there was an increase in the denervated limb. Now, they made traditional FSR calculations, even though this was decidedly a non-steady state. So we worked with a mathematician to develop some models for how do we account for non-steady state. And basically, this is a change in protein over time. Synthesis rates are best modeled as a zero order equation. They're not dependent on protein mass, whereas protein degradation is a first order and it's dependent on how much protein mass is there. And then we developed some other equations basically to calculate fractional synthesis rate to account for the fact that FSR would just go up as an account of losing mass over time. So we've developed all these equations, and I don't want to go into the derivation of them, but I want to show you how these equations impact our rates of synthesis that we calculate. And we did this recently. We reproduced this denervation model and looked at 7 days later using a D2O labeling during that period of time. And like the other study, we showed a decrease in muscle mass with denervation. Now, if we calculate the different ways of synthesis, if we just use the traditional measure of FSR, like the other study did, that doesn't account for non-steady state equations, we would have calculated a large increase in myofibrillar protein synthesis during denervation. But if we use our equations that we derive, say fractional synthesis rate that accounts for the change in mass, we actually show that there's no difference in the protein synthesis rate. And if we use the equation that does this based on protein mass, so in the case of FSR, this protein mass is less, and then this looks at the actual micrograms per time that we're synthesizing over that period of time, we see that there's actually a decrease. So in either of these approaches, we're not getting the same answer that the paper did that did not account for the change in protein mass. And what's important with these equations is we can also calculate protein breakdown. And if the other paper made a steady state assumption, I guess, when they made their calculations, so FSR would have to equal fractional breakdown rate. But when we calculate using our non-steady state equations, we see that there's actually a large contribution of breakdown to this change in mass. Now, if we look at the collagen pool, during that period of time, there's an accumulation of collagen, which is sort of a fibrotic response. If we use steady state FSR, we would have seen just a slight decrease in the synthesis rate. But since we can calculate synthesis and breakdown using our equations, you would see, again, a small decrease in synthesis. But what is really striking to us is that there's almost a complete lack of breakdown, which causes this accumulation of collagen over time, or this fibrotic response. Therefore, if we were trying to target fibrosis, say in skeletal muscle during a period of disuse, this one now appears to us as to be the more important side of the equation to target. It's not so much synthesis, but the lack of breakdown during this disuse period. So how can we use some of these approaches then to inform studies of intervention, or how can we apply some of these newer approaches to help us understand how to target specific conditions? So we've become very interested in this period of disuse, say during like a hospital stay. And the reason is because, as opposed to the general idea that you gradually lose mass over time with sarcopenia, with age, these periods of disuse become really important because in older individuals, there's a failure to completely regain this mass with disuse. Younger individuals or adults can regain this mass and go on. But with older individuals, there's an accelerated downward trajectory because we never really quite regain that mass after a period of disuse. Thus, these periods of disuse become a really important place to target. So what has traditionally been done in the literature is that there's been a really strong focus on mTOR activity. And this is taken from lessons that is mostly based on studies in young animals or young humans. By using strategies such as hormones or amino acid feeding to activate mTOR, it is thought that maybe we could increase this regrowth after disuse to mitigate this accelerated loss of muscle mass over time. So for these studies, we use a hind limb suspension model that is very well established. And in this particular study, we did adult and old rats. And you can see that in 2 of the muscles that the adults regain protein mass or muscle weight quite quickly within 15 days in the gastroc, and actually within 7 days in the plantaris. But the older muscle mass is very slow to regain and finally does by 60 days in the gastroc. But in the plantaris, it actually never regains mass by or at least by 60 days, it still hasn't regained mass. So we see what we see in older individuals in the rat model as well. Now, if we look at the weight-bearing period prior to hind limb suspension, and we look at the hind limb unloaded period, and we look at the reloaded period, these are all measures of mTOR activity. And what we see is that mTOR activity in older animals is actually higher than in the adult animals. So that got us thinking, well, let me support these findings as well. So if we look at the myofibrillar proteins that are the fraction nu over this 60-day period, you see that the adults in the blue here are at least as good or better than the adults at making muscle protein. And you see that here as a FSR by day 15, and fractional breakdown rates are higher as well. And if we look at RNA concentration and RNA biogenesis using our tracer techniques, the old muscle is very good at making RNA, so that's not the problem. And you see this again by the day 15. So clearly the older muscle is just fine at making protein and making the RNA to support protein in the muscle growth, but yet it's still not growing. And if we look at the soluble and insoluble fractions of these proteins, what we see is more of that protein is actually ending up in an insoluble fraction. So the making of proteins is not the problem in the older animals. It's actually that they're making it and not being able to process it into a form that is usable, basically. So for us, the strategy to prevent these periods of critical loss should not be working to activate mTOR activity, which is already high to begin with. To us, maybe the better strategy would to do something like inhibit this mTOR activity so that we're slowing the processing of the muscle proteins to prevent so many of them from being becoming insoluble or just being broken down right away. So I think some of these strategies that are activating mTOR are probably just making the problem worse. So important future directions in our lab. We're really interested in ribosomal biogenesis and ribophagy. We've noticed that or we have found that in this disuse period, we have a significant amount of ribosomal breakdown as well that we want to understand more. Downstream proteostatic stresses. So we want to understand some of the protein folding and how to prevent some of these proteins that are clearly being made from ending up in an insoluble mess. We're interested in the collagen turnover, fibrosis, and mechanical stresses, and how to improve this with age to minimize fibrosis. And again, one of the major directions in our lab is understanding of aging in vivo at the individual cell and individual protein level, so we can understand where to target our efforts within a tissue, which is a complicated series of cell types and individual proteins. So important takeaways: tracers allow us to capture a moving picture as opposed to a snapshot. The long-term nature of D2O labeling has increased our understanding of complex physiology, but the long-term nature also has special considerations. So it's not just about applying methods that we used to use using amino acid infusions to D2O. There's certain things we need to account for, but some of these things can be used as an advantage if done correctly. And I think these things can help us decipher mechanisms as far as like where to target our efforts as far as interventions. And right now, I just want to acknowledge these are the current members of my lab. This is the current primary support to the lab, and the various affiliations here at OMRF and OU Health Sciences across the street from us. Thank you.
Thank you very much, Dr. Miller, for a wonderful seminar. Very enlightening about sort of the current state of technologies to look at protein synthesis and degradation, and how sort of the simple sort of stochastic models that were sort of originally proposed, I think, obviously have some subtleties and limitations that it's always good to consider. There are a number of questions coming in. So let me go with the ones that are coming into the Q&A.
Okay.
The first one is, do you have label in the D and the O, or just the D? I guess that's water. And do you have to be concerned that you are seeing more or less rapidly exchangeable D?
So the deuterium is the only label we have. So we don't analyze O18. That would be a doubly labeled water approach. Some people have tried to combine, your traditional doubly labeled water energy expenditure bolus type experiments with protein turnover as well. But we are only focused on the D2. That equilibration happens very quickly. But when we actually analyze the samples for the product, we are not looking at the carbons that have labile hydrogens or deuteriums. So it can't just be jumping on and off into the water based on pH or other things. We look at the ones that only get in by metabolic exchange and then are sort of locked in there in the product.
Okay, the next question is, how is the turnover of extracellular proteins, collagen, elastin, regulated? What is the effect of caloric restriction on these proteins? Is it possible to facilitate degradation of damaged collagen?
That's a good question. And I'm trying to think back to all of our studies that have used either something like caloric restriction or rapamycin, whether we have looked at collagen turnover. I think nothing has leapt out at me yet, but I'd have to go look at that in more detail. I think that's an excellent question. Whether that increases some of these MMPs or proteolytic processes that would minimize fibrosis over time. I think that's a good question.
So there's a follow-up question. There's a very nice compliment that this was a great talk, and see the work directly from you. And it follows up on this. It's similar, and maybe there's some added nuances here. So I'll ask it. You showed that amounts of new collagen are relatively small in adults and decrease with age. Since collagen and other ECM proteins are extracellular, requiring MMPs and similar proteases for turnover, do you generally find that extracellular proteins are less likely to turn over than intracellular proteins, which have proteasomes and autophagy to assist in their turnover?
Yes. The short answer to that is yes. Extracellular matrix become much more resistant to turnover than we see in intracellular proteins. We recently did a study in cartilage as well that we have just recently submitted for publication, where we see that there's a large proportion of the extracellular matrix proteins that are actually completely resistant to turnover, and then some that do. But the intracellular proteins then are much more dynamic in that tissue as well. And one of the purposes of doing these individual proteins, to look at them individually, right now, one of the main goals of ours are to see which of the individual proteins intracellularly may become resistant to turnover with time. And the big goal of this, say if we look in the brain, is to think about things like plaques and tangles and stuff that get resistant to turnover. In muscle, we accumulate aggregates as well. So we're trying to understand what influences the individual proteins that may become aggregated over time, and that can happen intracellularly and extracellularly.
So Ben, I'll take the chairman's prerogative here and ask kind of a question that falls onto this, because I've always been I really enjoyed what you said about sort of many of the assumptions that go into assessing, metabolic kinetics with stable isotopes and assumptions around steady state and what the actual pool that's turning over is, et cetera. And I was wondering, just very generally, these proposed non-steady state equations that you have now developed and generated, do they account for those pools of protein that aren't turning over? And how does that factor in with this? Maybe they don't. I just was curious about how that gets considered.
They do. It's a good question. So the way when we do these, we'll do a time series. So a rise in enrichment over time, or a rise in fraction nu over time, more accurately. And then we'll look to see where those proteins are plateauing. If they are plateauing at a lower value, then we have to consider that within our non-steady state equation. But we haven't, we're just getting to that point of tackling this at an individual protein level, it's challenging.
I
But
mean, the other thing, just to think, I mean, just intuitively, those proteins, how much can a cell allow those proteins to accumulate without starting to degrade them? They can't just sit there. So,
so I think in some, it depends on the tissue. Like if you were thinking in muscle, no, they can't just sit there, and these aggregates become a big problem. If you look at something like extracellular matrix in the muscle, or we've done this in the liver too, in the muscle, I think there's a structural component to it. I think it makes sense that some of these extracellular matrix, these collagens, are not turning over. They create a sort of a structure that probably they're meant to. But I think in something like liver, when you're getting the accumulation of this extracellular matrix or collagens that are insoluble, that that has obvious problems then. So I do think there's some tissue specificity to it, as well as intracellular versus extracellular.
thank you. Are there examples where the same protein has different rates of breakdown or turnover in different cell types or tissues? What damages result in the cross-linking and insolubilization, and are these different in different kinds of cells?
Okay, so two very different questions there. So absolutely yes. You're going to have differences in a given protein in different cell types and different tissues. And that's why we have to look, we can't just say muscle is indicative of everywhere else in the body. So that's why we're doing brain and muscle studies, to start to get a grasp on how these differ between tissues. The second question, so how do these things, I think the question was, if they're insoluble, how does this lead to damage? And I don't think they're leading to damage. I think they become more susceptible to damage when they're in this state of not turning over. So then they accumulate things, non-enzymatic modifications, ROS damage, those kinds of things that then make these protein sort of structures accumulate damage, which I know AGEs and things can also trigger then further responses through rages and things like that. But yeah, I think they're both a cause and an outcome of damage.
I don't see another question yet, so I'll ask another question. So I think I really enjoyed the discussion you had about the idea that sort of basal levels of mTOR, I believe it was phosphorylation, are elevated in your aging animal systems. And I think we have some data in humans that's pretty similar, and probably some of our rodent studies too from a while back. But then, it gets complicated, because clearly if you contract muscle, you get, at least young muscle, you get a very robust increase in mTOR phosphorylation and many of the downstream kinases in that pathway, and it does stimulate protein synthesis. And I wonder if it's a little bit too difficult to generalize about, using mTOR inhibitors to sort of facilitate mass secretion. I just wonder how contraction kind of fits into sort of that model. I mean, maybe with atrophy, I think it could be possible, but
yeah. So it's a very complicated question, as I think you're recognizing as you ask it, that there's a lot of nuance to this. But we would like to target this period sort of leading in and during the hind limb unloading period with rapamycin. So not so much during the reloading period, where, like, as you acknowledge, the contraction might be important. But there are studies out there, David Glass and two other studies that I know of that have done rapamycin treatment over a fairly long period of time and shown a preservation of at least muscle function, better than without treatment. And then when you look at some of the animal models with chronically active mTOR activity or obesity with chronic mTOR activity, the muscle doesn't do very well. It does horribly. So there is a balance, and I think you're right that there's times where it's going to need to be up or it's going to need to be down. But that's why I'm sort of thinking about targeting periods rather than just always having it on board for long periods of time. Although, the long-term studies that have used rapamycin have shown quite beneficial effects on muscle.
Thank you. In lipids, deuteration near a double bond can prevent proton abstraction and PUFA oxidation. Does deuterium carry any caveats as opposed to, say, C13 in terms of controlling the rates of protein synthesis degradation?
So good question.
Last question too.
Yeah, and the levels of deuterium that are typically necessary to change biological processes are way higher than what we use. So in vivo, in humans, we target less than 2% enrichment in the body water pool. In rodents, we usually do about 5%. So you're thinking about 20 to 30%, I think, enrichment before you start having biological impacts. And it's at the enrichments we use, it's I can't say for 100% certain it's benign in all processes, but it seems to be benign in protein synthesis studies.
Thank you very much, Dr. Miller, for a wonderful seminar and Q&A here. We are now at time, and I'd like to thank all of our attendees and hope you can join us at our next Monday seminar. Thank you all.
Thank you.
Thank you, Dr. Miller. Oh, he's off. Okay, so I'm